So in this brief instructional video, I'm going to explain why meiosis is important to sexual reproduction. And I know this looks kind of a lot of information on this, but most of this you already know, and it should be pretty straightforward. So we're going to start over here, okay? And this, if you remember from when we talked about the difference between meiosis and mitosis, we talked about how in every single cell in your body, in your somatic cells, right? So body cells, you have 46 chromosomes. 23 of them come from mum, 23 of them come from dad, and they're in what we call matched pairs. So there are actually 23 pairs, but there's like 1A and 1B, 1A, 1B, and then there's 2A and 2B, 3A and 3B. And in each pair, one of those chromosomes started in the sperm cell that your father provided, and the egg, the other 23 were in the egg cell that your mother provided, and that's what gave you your full set. Because these are body cells, somatic cells, right? When you look at the chromosomes, they're in pairs, which means this is diploid, and because if this was a human, right, N would equal 46. There are 46 chromosomes in total. Okay, I should say that all organisms, not humans, have 46 chromosomes organized into 23 pairs of 2. But every other animal or plant or living organism, they all have different numbers of their DNA is split into different numbers of chromosomes. But within any organism, however many you have in your diploid cells, you have half the number in your gametes. So this would be a somatic cell. Since we're talking about the production of gametes and we're talking about meiosis, this would be a cell in either the testes, if it's a male, or the ovaries, if it's a female. Okay? And this is a cell that's, I'm going to say in the cell cycle, this would be G1. Okay? The cell's growing, it's doing its normal stuff. The next stage after G1 in the cell cycle, we know, is the S phase. And we also know that in the S phase, DNA makes copies of itself. So while I've still got the same number of chromosomes, you can see how they've kind of doubled in size. That's because all the DNA that's in the blue chromosome has made a copy of itself, and all the DNA that's in the pink chromosome has made a copy of itself. We are going to fast forward. We're going to go from the S phase, we're going to skip through G2, and we're going to go to prophase. Okay? In fact, this is called, because it happens twice in meiosis, but I'm not really interested in or worried about doing all the different stages, but this is something that happens during prophase 1. And if you look here, can you see how they're kind of like cuddling up, they're crisscrossing over? In fact, during prophase 1, we have something called crossing over, where chromosome 1A and chromosome 1B crisscross over. Chromosome 2A and chromosome 2B would crisscross over. So it happens between matched pairs of chromosomes. This is really, really important, because what they're really doing during crossing over is they are trading out sections of DNA with each other. And what that does is it produces variation. So anytime you see the phrase crossing over, you say to yourself, that produces variation. That's what makes each of us slightly different from our siblings, crossing over during prophase 1 of meiosis. The end result after prophase is this. And you'll notice now that there's pink bits on the blue one and blue bits on the pink one. And you'll also notice here how they're lined up in the middle. So this would be metaphase. Like I said, I'm not too worried about the remembering all the names. Other than prophase 1, where crossing over, you don't really need to remember all the different phases. But they line up, and then you'll see how each of them is separating, and then you'll see how that produces 2 new cells. I missed the pink bit there. Okay? And then fast forward these 2 sections of, so these 2 sections of this chromosome are going to move to separate ends, and these 2 sections of this chromosome are going to move to separate ends, and so this will end up in one cell and this will end up in another cell. Or alternatively, this part and this part could end up in one cell, and this part and this part could end up in another cell. What we have is something that we refer to deciding which bits go where. As long as you've got a top and a bottom part of this chromosome, it doesn't matter which ones. That is called independent assortment, which just basically means it is completely random which parts go to which cell. Okay? And what you'll notice here at the end is that we have 1, 2, 3, 4 chromosomes. Okay? I'm sorry, 4 gametes. So we have 4 cells here, and we started with 1 cell over here. Okay? And the other thing you notice is instead of these chromosomes being in pairs, they are singles. And the term we use for that is haploid. Okay? And so in each one of these cells, instead of n being 46, where you've got 23 pairs, 23 times 2 is 46, you have 23 singles. So 23 times 1 is 23 in humans. And these are all gametes. So if the original cell over here was in the ovary, these would be egg cells. If the original cell was in the testes, each of these would have sperm cells. It would have a tail. Okay? Now, I want you to notice something else. I want you to look at these cells. You will notice that first, not one of these has the same combination as the original one. They have some which are the same, but they're not completely the same. So they are different from the cells that the cell that produced them, and they are different from each other. So each gamete has a different combination of genes. They are different from the cell that produced them, and they are different from each other. This is called genetic recombination. Okay? Genetic recombination is when you have cells, each cell is slightly different. And the reason each cell is slightly different is because during prophase 1, we got crossing over. Okay? And I want you also to remember, I've only drawn one, like 1A and 1B. I've only drawn one pair of chromosomes. But this is happening in all 23 pairs of chromosomes. So it's not just these are slightly different. This is like where we got one. Imagine this 23 times. So 23 different chromosomes are all slightly different from each other. Okay? That's really important because, like I said, these are gametes. And gametes have one job. A sperm cell's one job is to swim and find an egg cell and fertilize it. So If this is the lucky sperm cell that does it, you're going to end up with one set of DNA, set of characteristics. If this is the sperm cell that gets to the egg, you're going to end up with a slightly different combination. If this is the sperm cell, so even though these 4 sperm cells all came from the same egg, because they are all slightly different, that means that any fertilized egg cell is going to be slightly different. So while you have, if you think about you and your siblings, you may have some characteristics which are more like your mum, some which are more like your dad. You may have some which are more like your grandparents. But not one of you has exactly the same combination. So there are all slight differences, and that's one of the most important things about meiosis. We have first, meiosis produces gametes. Then we have meiosis reduces the number of chromosomes so that when you have fertilization, you go back to the number you started off with. That keeps, each generation keeps the same number of chromosomes. And then you get crossing over, which produces these small variations in every one of the chromosomes, which give you small differences in characteristics. So once you've written this, what we're going to do is we're going to do a little, fold it in half, and we're going to write this. All right, 3 reasons, okay, why meiosis is important. Oops. We've got number 1. It produces gametes. You can't have sexual reproduction to produce offspring if you do not have sperm and egg cells, or if you're a plant, if you do not have pollen and egg cells. So meiosis, 1, it produces gametes. The second reason, it's called a reduction division, because not only do we end up with 4 gametes for every cell that produces them, but the number of chromosomes is reduced. So the chromosome number is halved. We go from cells having chromosomes in pairs, being diploid, to cells having chromosomes in singles, being haploid. So we go from a pair of chromosomes to chromosomes which are in singles. That's another really important reason why meiosis is important. And the third one, really important for why meiosis is so critical, is, right, crossing over, and then that kind of like lottery about, which part ends up in which cell. So crossing over and independent assortment, those two things produce genetic variation. Why you are similar but not identical to your siblings. And I'm sure you can think of things, if you have more than one sibling, you can think of things where you've got something that looks more like dad and they've got something that looks more like mum, but then you've got maybe all got the same color eyes. So why is meiosis important? 1, it produces gametes. 2, it makes your chromosomes go from pairs, diploid, to singles, haploid so they can go back to being diploid after fertilization, and then crossing over during prophase 1 and independent assortment lead to genetic variation.